Alright, hello. My name is Jackson McQuaid, and this is my Biology and Concepts in Biology 101 course, Project 2. And my protein is Piezo1, and the mutation I was assigned is that arginine 2456 is replaced by histidine R2456H. And so, three limitations of this, this model, and the Play-Doh I made it out of, is that the model isn't able to show the ion channel actually opening or closing for any of the ions. It is extremely difficult. Well, it isn't extremely difficult, but it was difficult to show the mutation because it's just a replacement, and it only shows the shape when viewed from top down, as I couldn't get it into a conical shape as it is viewed from the side. All right. So then, what is the normal function of this protein? Categorize the type of function this protein holds. Well, Piezo1 is a receptor for mechanical signals, specifically stress, and it reacts by opening an ion channel in response to stress. It is specifically important in embryonic endothelial cells because these cells with Piezo1 are shown to more accurately change the alignment to suit the flow of red blood cells more than those with less Piezo1 in the system. And embryonic endothelial cells are cells in an embryo that develop to form the cells along your veins, so not the actual blood cell itself, but the thing that is containing it. All right, so then it has to be receptor specifically because it's an ion channel, and that is obviously one of the three main, receptor proteins, but it doesn't break bonds, it's not important structurally, it doesn't transcribe RNA, and it doesn't transfer a signal, but it does respond directly to pressure, and this makes the protein a receptor protein. The impact of this protein is really mainly shown in the aforementioned embryonic endothelial cells, but it is present in most other cell types, but it is expressed in other cell types by allowing cells to sense changes in body fluid pressures. It is also important in red blood cells as it allows for these cells to maintain homeostasis, so it lets calcium in or out of the red blood cell so it doesn't shrink or burst, and that really keeps you alive, so it's very important. The impact of this protein on the organism is that if not enough Piezo1 is present in developing embryos, the embryo will most likely die about halfway through development due to lack of blood flow, or die because red blood cells could not maintain homeostasis and became ineffective, either preventing the transport of oxygen or just bursting entirely, killing the embryo. And then where is this protein normally found? Subcellularly, Piezo1 is found in the plasma membrane of eukaryotic cells, and it is widespread through eukaryotic cells, present in lung tissue, blood cells, the nervous system, bladder, and skin cells. And it is shown in almost all eukaryotic organisms, ranging from current humans to prehistoric creatures like dinosaurs, but it is never present in prokaryotic cells like bacteria. And then categorize the type of change this mutation has had on the protein function. Explain why you chose this statement in relation to how the mutation works. And I chose B, the protein becomes more active. I chose this because the Biol101, one of the sources that was provided, I think it was the first one, says that the mutation causes Piezo1 in red blood cells to become more active, and as a result allow more calcium into the red blood cells, and create a three-dimensional model. Oh, it's already on there. Yep. All right, so what are the main functional groups present in the original amino acid side chain? The functional groups in side chain were a carboxyl group, an amine group, and with a hydroxyl group on the carboxyl group, with a guanine group in addition to all that. And then discuss the properties of the original AA side chain. The original AA side chain amino acid side chain is very hydrophilic, and it is considered one of the most hydrophilic of all the natural amino acids. And then, is it neutral, positively, or negatively charged? It is positively charged, and this is due to the fact that the side chain is basic and it has to pick up a proton, become positively charged. How would you characterize its size compared to the 19 other standard amino acids? And it's quite large compared to almost all other amino acids, but it is still smaller than a few of the others. And then discuss the interactions that the original amino acid can participate in. And I said because arginine is polar and it is basic, then it usually just spends its time at the cell membrane, and the amino acids will develop so that the hydrophilic amino acids like arginine go out towards the cytokinesis and the extracellular area, while the hydrophobic parts that don't really have too much arginine in them will stay in the middle of the cellular membrane. And then, what are the main functional groups presented in the mutated side chain? Well, the main functional groups presented in the new side chain are an amino acid group, an amine group, and a carboxyl group with a basic group, and the basic group being an imidazole ring. Discuss the properties of the new mutated AA side chain. The new mutated structure is neutrally charged, so it ends up being nonpolar. The new mutated structure is neutrally charged, so it is neither positively or negatively charged. And then, how do you compare its size compared to the other 19 standard amino acids? I would say it's slightly above average inside, but it is still smaller than the original amino acid arginine. And then discuss the differences in the types of amino acid. Because the new amino acid side chain is acidic and nonpolar, it can go into areas that the original membrane was not, and it also opens up the ion channel more easily and such like that. How does your side mutation affect the protein function, the function of the cell, and the function of the organism? This mutation causes the protein to become more active, and this in turn brings more calcium into red blood cells. And this would be negative, but in some parts of Africa where malaria is common, this mutation is very, very helpful because it causes red blood cells to shrink, and this can avoid malaria. And as a result, the gene mutation is significantly more prevalent in areas that are affected heavily by malaria. And of course, this is the arginine, this pink dot right here. That is supposed to represent where the mutation occurs. And because there's no visible difference in the protein, then I just kept with one model instead of making a exact replica. So